Thank you very much, Dr. Thank you very much for the invitation. And eminences and brothers and sisters in Christ, we have heard a great deal about what technology can do, and I need to address the dangers of the technology because by addressing the problems of the technology, we will be able to oppose much of what is being proposed in altering human behavior. The fall of man is the intent to go beyond what is possible for a creature to become, that is, divine without God. And reconciliation, of course, with God is a normal state of creation. And the problem then is ontologic. It's the failure to accept death as a natural end of earthly existence and a divinely permitted corrective to fallen human nature that leads to nihilism. In the search for amortality, not immortality, but amortality, aging becomes a treatable disease. Now, aging is a result of a highly interconnected network of biological cascades resulting in the degradation and breakdown of every living organism over time. Anti-inflammatory agents, oxidants, plasma infusion from young people, embryonic stem cell infusions are all interventions to delay aging. Botox, aesthetic surgery to maintain a youthful appearance. Because disease then is not viewed as a result of lifestyle choices. We ignore AIDS, obesity, emphysema, and certain cancers as examples, because vaccination and pharmacotherapy displace the need for self-reflection and correction. One overcomes the genetic and epigenetic limitations of the creature with performance-enhancing drugs, bionic appendages, which are not related to any medical need. I, for example, have a pacemaker. That's quite a different situation. But a future as a cybernetic android is really unlikely for two reasons. One is that's the physical limitations of the organism, and secondly, of course, are the energy needs in order to maintain that state. So let me talk then about genetic engineering, because that is the ultimate affront to the Creator. It's the challenge to the very nature of the human being and his inherent image of God, because it is man who chooses which genes to edit. And the principal goal of this, of course, is to create a Superman. To create the Superman, or Ubermensch, implies a recurrent source of compatible organs to facilitate continual replacement of aging systems. The clone, then, is defined as not being human. It cannot be human. It does not have a soul, and therefore is not deserving of any protection. The definition then of what is human becomes that of the exploiter and the usurper. I've discussed this at length elsewhere. This can be downloaded from my website or purchased, but it's certainly available. The historical experience of the unintended consequences of animal breeding are forgotten. We forget that before the sheep Dolly was cloned, there were 277 failures that were discarded. Thus, a brief review of the essential techniques of genetic engineering will illustrate the devastating consequences that occur in humans, who at the moment cannot be discarded. And knowledge of the process will enable an informed opposition to this hubris that is genetic engineering. The genetic code, as we've heard before, uses a four-letter nucleotide alphabet. During DNA synthesis, the DNA double helix is unwound. The strands are copied. A DNA polymerase proofreads the copied strands to repair copy errors. There are at least 5 major repair pathways active throughout different stages of the cycle, and 2 of them repair double-stranded breaks. One of them, and hopefully tempting to share this slide. Well, in any event, one is homology directed repair, and information from the homologous chromosome that matches the damaged one is used to repair the break. In this process, the two homologous chromosomes come together, and the undamaged portion of the homolog is used as copied, is used as a template to repair the damage, and the chromosomes are joined together. This is a very precise correction, which is why we use it in genetic engineering. It rarely causes mutations, and we can insert or delete or change genes very easily. The second is called non-homologous end joining. Now, the two broken ends of the chromosomes are just simply glued back together. The repair typically involves insertions or loss of nucleotides at the cut site. It cannot induce precise mutations. It does not require a template. It occurs in both dividing and non-dividing cells, whereas the homologous end joining, homologous recombination only occurs in dividing cells. But the final outcome in this process depends on the repair, because frequently induces what's called a frame shift. For example, 3 cytosines code for the amino acid proline. It's nonpolar. If there's a frame shift and I have adenosine, cytosine, then I get production of the polar amino acid threonine. So I've changed the protein and character of the protein and the electrical characteristics of the protein in this episode. The CRISPR system that we use comes from bacterium archaea, and it was a defense mechanism in the bacteria to distinguish invaders from the host. And it's a system that's designed to cut DNA in specific places. And the system consists of single-guided RNA that creates a double-stranded DNA break at a specific site. And the site is called a target sequence. And the simple pairing of complementary nucleotides leads the attached enzyme to the target sequence. There's a specific 3 to 6 base nucleotide sequence directly after the target sequence that's recognized by the Cas enzyme and triggers its activity. If you don't have this nucleotide sequence, there's no cutting, there's no activity, no editing. The CRISPR-Cas system then is primarily used to induce these site-directed double-stranded DNA breaks. And the actual genome editing then occurs in the reparation of that break. The system is programmable because we can add additional enzymes, we can insert genes, swap out genes, silence genes. And so it's a very effective tool for gene editing. It can be delivered into the cell using a vector, either bacteria or a virus, a ribonucleoprotein complex, or nanoparticle. In an ex vivo strategy, the genome editing takes place in isolated patient cells, and the engineered cells can then be expanded to be reintroduced into the patient. The advantage is that the editing occurs on a cellular level, so it can be assessed clinically before being delivered to the patient. Any non-homologous end joining errors can be removed. The CRISPR system is not introduced into the body. It decreases the possibility of any changes to the genome of non-target tissues. And finally, as they're patient-derived cells, there's not a possibility of immune rejection. In the few cases where this has been attempted, the results have been disappointing. And that's with HIV and cancer therapy. And in some instances, such as treating beta thalassemia or sickle cell anemia, there have been some significant adverse consequences. There's a small ongoing program today to attempt to treat muscular dystrophy. But again, these all involve ex vivo strategies. With an in vivo strategy, genome editing takes place inside the entire organism. And so it's possible to alter the genetic population, the genetic structure of an entire population. In vitro transcription uses a bacteriophage polymerase to produce a long transcript of RNA. RNA is what is read that generates the protein production inside the cell. It's generated from the DNA template, as we've described above. It was recently used in the massive gene experiment known as the COVID vaccine. In editing the genome, the nucleotide sequence is optimized. It's optimized to maximize protein production, the proteins that one is interested in. It's based on the fact that specific organisms prefer specific nucleotides. However, this optimization can lead to a change in the multifunctional regulatory and structural roles of the resulting proteins. That is, they have different electrostatic characteristics, they fold differently, and they have different conformation. And so these problems are seen and can be seen. So these off-target proteins, because of these changes, may be recognized as foreign by the immune system. They may not be cleared by the body, and they may lead to multi-organ damage. The use of synthetic nucleotides, such as N1-methylpseudouridine, in generating the mRNA, because it adds to the stability of the product, however, increases the rate of translation errors. And further, it impairs signaling pathways in the innate immune system. It blocks p53 activation, which is a key checkpoint in the cell cycle. Viral infection is not well addressed following treatment. Foreign antigens are not cleared. And so autoimmune disease and cancer progression are expected and have been described. A plasmid or vector is employed for the delivery of the mRNA in vivo. The delivery permits the cell to produce many copies of the delivered mRNA. The vector comes from a bacterium or a virus. That genome also incorporates in the genome of the target organism. The adenovirus, cytomegalovirus, and SV40 virus vectors that we use are oncogenic. The SV40, particularly as was used in the COVID vaccine, is known to affect a somatic hypermutation, that is class switching in B cell lines, so it's not surprising that we're seeing an increase of lymphatic cancers in vaccinated patients. To avoid recognition by the immune system of the host, the plasmid is coated with a lipid nanoparticle. It's taken into the cell. Once into the cell, the lipid nanoparticle is released, damaged. It damages the cell, causing inflammation, but it releases the mRNA. So the adverse effects of the gene therapy experiment that has been undergone worldwide, which was the COVID vaccine, they're substantial and predictable and are ongoing. For example, the Cleveland Clinic points out that anyone who has had myocarditis by this vaccine, roughly 50% will be dead in 5 years because of the vaccination, because of this gene experiment. So then let's talk about cloning. Because cloning of the organism is best accomplished utilizing an embryonic stem cell. However, we can reprogram somatic cells to an earlier state, but it's easier if we remove the nucleus from the egg. We put the nucleus from the cell of the organism to be cloned in the egg, and then it's stimulated. Of course, if we take the donor egg from the mother of the organism, then it's a true clone because it will be translating the mitochondria as well.
Father,
and using CRISPR-Cas system,
Father, you
we can
have 2
edit
minutes
the
to
embryo,
kind of
the embryonic
summarize.
stem cell in vitro, and then the failures are discarded. Well, that's what's done with in vitro fertilization. The embryos are discarded. That's, of course, a significant ethical problem. But in any event, and today there is, in fact, a UK company that's offering to screen such embryos to permit such genetic editing. But more disturbing is that human germline epigenetic reprogramming in vitro has recently been described bypassing the need for sperm or ova. Patents have been filed by the California Institute of Technology and Cambridge Enterprise Unlimited, as well as the Weizmann Institute for creation and sustenance of synthetic human embryos. So in the end, the alteration of the genome of an entire population has already occurred with significant adverse consequences. It is possible using these techniques to actually select a gene to be edited which has differential expression in certain populations, so it can be used for biowarfare. Animal cloning continues. We're even speaking of...
Father, you need to summarize your presentation in the next 2 minutes, please.
Right. In any event, the point is that regulatory bodies are captured by the commercial interests. We are here to witness to the truth. If we do not move to stop or alter or oppose this hubris, this is not going to end well. And thank you for your attention.